Bismillahirrahmanirrahim. Dear students, now I am going to explain the monoclonal antibodies. The term mono means one type. As you know that mono term is used for the one type. Here is a misconception among the students that they consider that monoclonal antibodies mean that it can produce only one type of the antibody for a specific antigen. So I am going to explain the term monoclonal today. Mono means one type, and as in the previous video we discussed that we hybridize the spleen cells with the myeloma cells. We combine spleen cells of mice with the myeloma cells, which are cancerous cells, and by the combination of spleen cells and myeloma cells we prepare the hybrid cells, which are called the hybridoma cells. And these hybridoma cells have the characteristics of spleen and myeloma cells. That is already discussed in detail in the previous video. Now let's suppose we have hybridoma cells in our body, and it sends certain type of the antigens. Let's suppose the antigen is HBV, that is HCV, or that is bacteria. For all such antigens, the hybridoma cells will start cell division and they will produce new cells of their own type. Now the main topic is here. The parental cells, which are the actually hybridoma cells, and the newly prepared cells, which are the clone cells, they are 100% same. That's why they are called monoclonal antibodies. The monoclonal term is used here that the parental and daughter cells are same clones. Now these clones have the capability to produce variety of antibodies. Let's suppose they have since the HBV, so they can produce antibodies against HBV. If they have since the HCV, they have the capability to produce the antibodies against HCV. If they have since the bacteria, the clone cells have the capability to produce antibodies for the bacteria. The difference is here that the type of the clones are same. The type of the clone cells are same for all cases, whether that is for the HCV antibodies, HBV, HAV, or bacteria, or any other type of the antigens. In our natural immune system, the case is different. Here, the parental cells produce variety of clone cells. The clone cells for the HBV will be different from the HCV. The clone cells for the HCV will be different from HBV. These two will be different from the HAV, and the clone cells will be different for bacteria. But here we emphasize that the copy or the cells which are the clones, they will be the same. Monoclones mono means one type of cells will be there, but these cells have the capability to produce variety of antibodies. So because of this property, same clones formation, the term is used monoclonal. Monoclonal antibodies are used for two purposes. They are used for the treatment, therapeutic purpose, and that is also used for diagnosis. Treatment means that you can inject the hybridoma cells in every person's body, and they have the capability to replicate and produce antibodies in that particular human body and kill the particular antigen. So you can use it as the treatment source. You can also use it as a diagnosis. If it cannot kill the pathogen, at least you can isolate the antibodies from the blood of the patient, and you can recognize whether the person is infected with the certain pathogen or not. So monoclonal term is used for the clone actually for a type of cell. It is not used for the antibodies actually. Antibodies can be in broad range, it can be broad spectrum, but clones of the cells will be the same type. Thank you so much. Keep watching our channel for further videos. Inshallah, upcoming video will be about the diversity in animals from the first year, inshallah. Thank you so much.